Hi, I'm John Heymach. I'm the chairman of Thoracic and Head and Neck Medical Oncology at MD Anderson Cancer Center in Houston. So the question is, how does resistance arise to RET inhibitors like selpercatinib and pralsetinib? It's first important to describe how these drugs actually work in the first place. So these drugs are called TKIs, or tyrosine kinase inhibitors. And these are molecules that fit into what we call the kinase domain of a RET fusion. The way a RET fusion works is RET is a kinase, or it has a kinase in it, and the kinase part fuses with another gene like KIF5B, and that is a fusion, a KIF5B RET fusion in that case. And when that fusion happens, it sort of turns on the RET kinase, and that makes the cancer cell go. And so what a RET inhibitor does is it fits into that kinase domain, and that drives the tumor. It makes the tumor keep dividing. Normally, RET gets turned on and off in a normal cell, but in a fusion, the RET is turned on all the time. The term is constitutively active, meaning it's not getting regulated to get turned on and off. It's always on. So the RET inhibitor shuts off the RET kinase and the cancer cell dies. So how does resistance happen? Well, one thing is that the RET kinase can change. Sometimes it gets additional mutations, and when that happens, the kinase inhibitors can no longer fit into that pocket. So you can think of it, if it fits in like a lock and a key. Well, if the lock changes a little bit, then the key would no longer fit. The key being the kinase inhibitor in this case. Now, when that happens, we can try a different kinase inhibitor. As an example, there's a drug from Ellipsis, a RET inhibitor, and that inhibitor will inhibit some of the resistance mutations that can cause resistance to selpercatinib or pralsetinib. So if the resistance is because the RET gene has changed, then our strategy might be to try a different RET inhibitor that's able to inhibit the new mutated kinase. Now, there's other ways that resistance can happen. Sometimes RET continues to be present, but RET doesn't change. Another gene gets turned on. An example is MET, M-E-T. It sounds like RET, and it does a lot of what RET does, but it's a different gene. Now, if another gene like MET gets turned on, then you can block RET all you want, but you need to also block MET for it to be effective. We call this a bypass mechanism of resistance because a different gene is bypassing RET. So in that case, you need to target both MET and RET. You need to target both RET and the whatever the bypass mechanism is. Now, we think there's a lot of different bypass mechanisms. HER2 might be one, EGFR might be one. We think there's a number of different ways you can get resistance in that case. And that's where it's important to get a biopsy, see if a bypass mechanism can be identified. And if so, then devise a combination that targets both RET and the bypass mechanism also. A third sort of resistance is what we call lineage change. And this happens with all our different kinase inhibitors. With lineage change, the cell changes its basic phenotype, we call it, the pattern that the cell adopts. Most RET fusion lung cancers are adenocarcinomas. And adenocarcinomas are the type tumor cells that are often very dependent on kinases. But sometimes an adenocarcinoma can shift to a squamous carcinoma, or it can shift to a small cell. We call that small cell transformation. Or it can shift and undergo something called EMT, epithelial to mesenchymal transition. And the cells sometimes can be a mixture of these different lineages. When that happens, the cancer cell is not dependent on RET signaling in the same way as it is when it's an adenocarcinoma. And that requires entirely different treatment strategies. And that might be where we need chemotherapy or antibody drug conjugates because the cell is now indifferent to RET. So cell doesn't really care if you inhibit RET or not. It's got a whole different type that isn't dependent on RET signaling anymore.